Transcription is a process used to convert DNA to RNA. This happens in the nucleus, where RNA is defined as a picture of a small section of DNA. There are 3 steps in transcription. These steps are initiation, elongation, and termination. Before the first step, there was an enzyme called helicase, which opens the DNA so the RNA could copy. In the first step, initiation, the promoter DNA signs where and when the transcription starts, and also identifies which section of DNA should be copied. The next step is elongation, where the RNA transcript copies the non-template strand, but replaces the T's with the U's. The final step of transcription is termination, or the section where DNA tells transcription to stop the RNA transcript, and the RNA transcript splits and leaves the nucleus, which ends the process of transcription and the start of translation. Translation is a process that uses RNA blueprints to create and align amino acids to create a protein. RNA blueprints contain the information needed to build the final product, which is the protein. Before we get started on translation, we need a brief review of the codons. Amino acids are the building blocks of protein. Any 3 pairing of RNA nucleotides will code for a specific amino acid. Now back to translation. We have a ribosome. A ribosome is where translation takes place. It can read 3 tRNAs at a time. The ribosome reads the RNA strand until it finds AUG, which is the start codon to tell when protein photosynthesis begins. So there's many tRNA carrying amino acids. On each tRNA has an anticodon. Every time a new tRNA comes in the ribosome, the empty tRNA leaves. The tRNA in the middle of the ribosome carries all the amino acids, which is the growing peptide chain. When the ribosome reads either UAA or UAG or UGA, that means to stop the protein synthesis. These do not code for amino acids. After the stop codon is read, then the peptide chain splits and creates a protein.